Okay, we're going to stick with the same theme. We're going to talk about clinical data, human clinical data. So exciting. This is where we all wanted to be. 5 years ago, we couldn't even imagine dreaming to be here. So I hope you guys are super excited and really thrilled with what you're hearing today. Next, we're going to talk about another antisense oligonucleotide that's being advanced by Ultragenyx Pharmaceuticals. We have Dr. Kim Goodspeed, who I want to welcome to the community. Kim has been a part of the neurodevelopmental community for a long time. I've actually known Kim for quite a few years, mostly because we would speak on panels together. But she would generally speak about different disorders while I would speak about Angelman syndrome. So I've gotten to know Kim very well. And when she came to Ultragenyx, it was just such a pleasure to be able to have somebody in the field, a child neurologist, who truly understands individuals living with Angelman syndrome, because she's been treating them in her practice for a long time. So Kim, I know this is your first FAST meeting, and I know this is your first soiree this year into Angelman syndrome, but we are so happy to have you, and we welcome you and hope you're here for a really, really long time, because you're fantastic. So Kim is going to talk to us about GTX-102, the GTX-102 clinical development update, results from the phase 1/2 open label study, and the overview of the upcoming pivotal phase 3 study. Kim.
Oh my goodness. It's such a pleasure to be here. Y'all are a really warm and welcoming community, and it's truly been an honor to be in this room hearing all the amazing science that y'all as a community have put forward. So thanks for welcoming me in. As Allison mentioned, I was a child neurologist, I still am. Neurodevelopmental disabilities is my specialty, and I had been caring for kids with Angelman syndrome and other genetic disorders that are very similar to this condition for years. Frustrated alongside parents when I'm telling them speech therapy, physical therapy, here's maybe some medicines to help with different symptoms, but nothing's really moving the needle. And then to see all of these amazing opportunities coming forward and all of the just incredible science that has gotten us to this point. And now we're in clinical trials, seeing the trajectory of these diseases change. And it's just incredibly exciting. And I had to join the team to be on this side, being able to advance things and do my part to hopefully get different treatments in the clinic so that your future medical students learn a totally different story about Angelman syndrome. So, with that, I'm between you and a break, and I understand that. So we'll get through our data, and I'm really just excited to share this story with you. But the story starts by thanking you for allowing us to work with you as a family, trusting us with your children, and partnering with all of these amazing clinicians across the globe who have made all of this data possible. I just get to tell you the story, but y'all are the ones that made it all happen. So thank you for all of that. So I do have to give you some disclosures, right? I have to remind us, we have to level set, we have to understand what we're looking at before we go and look at it. GTX-102 is our antisense oligonucleotide, our ASO. It's not yet approved. So everything we're showing you today is based on an investigational product, right? And that means we're also showing you open label data, which I think you've heard a couple of times now, and y'all are gonna be clinical developers by the end of this weekend. And so that means that this is open label data. Everybody knew they were getting drug. We potentially can have some biases, both from families, potentially us as sponsors, as well as your clinicians. So we always look at the data with open eyes. And we have also included some quotes from the community, from some of our participant parents. And I want to make sure that we understand we're not guaranteeing that this would be every patient's experience, because each of your journeys is unique. However, we did want to bring the data to life a little bit and give you some real-world examples of what patients are seeing so that we understand when we're looking at graphs of Bailey data again, what does that actually mean? How does that look in a real-world setting for these particular individuals who have shared that with us? And first, what's Ultragenyx? So it was founded in 2010. It's a global company, about 1,300 employees, and it was expressly designed and developed with the rare disease community in mind. That's all we do is rare disease. Being an academician, looking for an opportunity to work on clinical development, all I wanted to do was rare disease, and this has been a dream to be able to work with this team that is so dedicated and patient-focused. We learn from you as a community. We incorporate as much of that as we can into our plans, and we really center and inform our decisions we make as a company based around what is good for the patients, what is good for the community. So GTX-102, it's an ASO. Y'all could probably teach me about ASOs at this point. I think you've been told about them so much. But ours, quick level set, we are looking at deletion patients to start. And so in the case of a deletion, we know that it's the maternal copy of UBE3A that's not able to make the UBE3A protein. And we also know that in brain tissue, the paternal copy, dad's copy of UBE3A, is silenced by this antisense transcript. That just happens naturally. GTX-102's job is to go in and silence the silencer, so knock down or silence that antisense transcript that relieves the inhibition of the paternal UBE3A copy, so dad's copy is now able potentially to make UBE3A protein, thereby, resupplying what has been lacking from mom's copy being unable to make the protein. So what we've observed in our phase 1/2 study, we're going to go into more detail, but because GTX-102 is a targeted treatment, it's getting at the core of Angelman syndrome, it is potentially going to have a different impact in different patients across some of those core features. Well, we got to learn those core features from you as a community because you've done decades of natural history work. You have disease concept studies. We get to learn from all of that, leverage that information, and really tailor how we design our trials to make sure we're measuring the right things. And so we focused on kind of 5 key domains: cognition, so learning and memory, thinking skills, problem-solving skills; communication, how you listen and understand language following directions; hyperactivity, challenging behaviors that make it difficult to go out in public or to do big family events or even maybe make it through dinner; sleep; and then also balance and coordination, being able to get around their world and be safe. And again, GTX-102 is not an approved product, but we are going forward with a phase 3 study we're really excited about. I'm going to talk to you about that at the end of our time today. And that will be our study that helps us really understand that safety and efficacy profile for GTX-102. So I'm so glad to be able to follow on the heels of all the great talks this morning. And this is just a roadmap of where we are. So we've done our phase 1/2 study. We enrolled 74 participants, and that completed enrollment the end of December last year, about a year ago. We've continued to follow those patients as we're preparing for the next steps, and so now we're here. We're ready to launch Aspire. Our plan is to open it by the end of the year, and we're very excited. The plan is for that to have about 120 participants. We also have another study that we are planning to open in 2025 called Aurora, and it is going to study those other genotypes, mutations, UPD, ICD, as well as the other ages that we have not included in our phase 1/2, so the younger kids as well as our adults. And that's going to open in 2025. So once we have enough data together, the next step, right, is we go to the regulators and we say, here's what we found. This is what the safety looks like. This is how we see it benefiting patients. What do you think? And we negotiate and we work to make sure that this gets to be approved, and that's our goal. But it takes a process, it takes time, and it takes partnership with you and communities like FAST and ASF to make all of this happen. Once it's an approved product in the future, it would go and be available to be prescribed clinically by your doctors at home. And then we continue to monitor safety and efficacy because we always want to learn and understand what's happening with these new treatments. So with that, we are going to talk about our studies one more time. So our phase 1/2 study, first time we went into human patients. We've studied it in deletion-positive patients, ages 4 to 17 years old. That study's completed, and they're all rolling into this long-term extension study. You'll see that this is kind of our landing page, our landing home for all of our clinical trials. They will all end up in this long-term extension study. This study opened up over the summer, and all of our phase 1/2 patients are transitioning over to that currently. Our phase 3 study we'll talk about in more detail towards the end. And it's our study where we're going to confirm the safety and efficacy. So that's our real goal of that study. Again, it's going to open by the end of the year. And then Aurora, just mentioned, is our study to look at it in other genotypes and other ages so that we understand its broad implications, because we believe it can work across the spectrum of patients with Angelman syndrome. And so we're studying it in this particular study to understand it in the other populations. You will see 3 of these studies are up on clinicaltrials.gov right now. So our phase 1/2 is up there in completed enrollment. Our phase 3 is up there, not yet recruiting. We'll update that as soon as we have more information. And as we have more information, this is going to be a really good source of truth for you as a community. And I'm so glad that y'all had that education and kind of guide to ct.gov this morning. Our long-term extension study, you'll notice, is by invitation only. And that's because it is pulling in patients from our other clinical trials. It's not to exclude anyone, it's just that that study is designed to accept patients from the other studies. All right, so now we get to dive into the data. This is sometimes the boring part of talks, right? You just see a bunch of graphs and things, but I'm gonna try to make it a little bit as lively as I can. So our phase 1/2 study was really a learning study. It was an opportunity to see what doses are tolerated, what doses are effective, and how do we want to structure our doses as we move forward with the program. And so our cohorts 4 through 7 and A and B, these are our dose escalation and dose expansion cohorts, have really given us a lot of information that have given us confidence as we're moving into our phase 3 study. And so those are the cohorts that we're going to focus on for today. We also have the longest term data on those cohorts. And again, talking through some of these symptoms, so cognition, communication, sleep, behavior, and motor function. And of course, we're always going to talk through our safety profile. So to review our baseline characteristics, it's important to know, what do patients look like when they enter a study? How similar are the different groups, and how close are they in terms of their demographics of age and gender, and their baseline scores being relatively similar is really important to understand at the beginning. And largely, cohorts 4 through 7 and A and B are all pretty similar across all these aspects. And then how do you understand the data you're seeing in a trial, especially the first time you're looking at it? And we're an open label study for our phase 1/2, and so that means that we don't have that comparator arm. So we used your natural history study data that your community has spent decades working on, volunteering your time, working through countless hours and countless numbers of assessments of the Bailey and Vineland, which I know is not easy. But it's so valuable because it gave us context for our study. It gave us an understanding of, what are the differences we're actually seeing, and do we usually see those changes in patients with Angelman syndrome who are treated with routine standard of care that's available now. So we've used both of these studies, but we've pulled out the populations that are similar to our population we're studying in the phase 1/2. So that means that of all of the patients that were in those natural history groups, we only pulled out to look at those ones who had a deletion genotype with the 4 to 17-year-old age range, so that we were trying to compare apples to apples as best we could. And so we're going to start with our cognition data. So Bailey cognition is such an important foundational assessment tool. We know the Bailey's not a perfect tool. We've learned that from Dr. Barry Kravis. I knew that coming in when I was in development myself. But it's the tool we have, and it's the tool that we can use right now to understand, what did y'all look like in the natural history study? What did these kids look like? How did they grow? How did they change over time using this tool, understanding all of its potential flaws? And how can we see that it changes when you give a patient GTX-102? So we've been showing you GSV scores. So that's what you see on the left, the blue bars. Growth scale value scores are a way to transform just a mathematical equation to go from a raw score on the Bailey to a score that is more linear and a little easier to interpret and compare across different studies and across different populations. And so what we see is that we're surpassing that dotted line. That dotted line represents what the Bailey manual tells us is the statistically significant threshold. So if you're above that line, it probably didn't happen by chance alone. So that's how we interpret this data. We also know from the natural history data that the score changes over about a year are usually less than 1 point, and we're seeing score changes of about 6.7 GSV points. So we're really excited about this data. But not just that, that's data at 1 year. We also see continued improvements past a year. So out to day 506, we're seeing continued growth with exposure to GTX-102, and we're just thrilled to see this. So our red bars are up here because we have to work with our regulators. We have to be good partners with our community as well as the bodies that control whether these drugs get approved or not. And they really prefer to see the raw score. So we took a look at it. What does it look like if we look at this using the raw score or the GSV score? And you see that the magnitude of change is really pretty similar. There's not a dotted line on that graph because there's not a statistically significant threshold that's defined when you're using the raw score. And so we'll be using minimal important differences. So an MID, I think we've talked about those over this weekend as well, to make sure that the changes that we're seeing are changes that are impactful for you as a community in your lives. And then the bottom tables are really just to show you that we've done the work to understand when we're looking at this as our primary outcome measure, do we think we've got a good chance of having a successful phase 3? And we say yes. We are well powered, so that means your ability to detect a change if the change is truly there. That's what a power calculation does for you, and we're powered at over 90% with both of these measures. So all the sciencey jargon aside, what does this actually mean? What do kids look like when their cognition scores improve? And it means that they're able to engage. We've heard from families that they are more attuned, more alert, a little more responsive, able to sit through a Zoom class, sitting through their therapy sessions a little bit better. So that foundation of being able to learn, commit to memory, develop new skills is really rooted in an improved cognition score. But we want to look at everything else, right? Because Angelman is not a monolith. You have lots of different symptoms, and each patient's journey is unique, so we have to understand how it moves on all of these different symptoms. So communication, being able to understand what is said to you, process it, and show that you've understood it and respond to it. So we know that on the receptive communication natural history data, you get about a 1 to 2 point change in score over a year. And in our left line graph is where we see our GTX-102 treated participants. And at a year, they're surpassing that statistically significant threshold above 6. And so we're really excited about this. And again, it's families saying, a major gain in comprehensive communication. Whatever we're saying to her, she understands it. And that can be so meaningful to families and really impact how they're also learning and participating in their therapies. We've also looked at sleep and a number of other symptoms. We're using the ASA, so the Angelman Syndrome Severity Assessment Tool. This was a tool designed by some key members of your community that are really anchored in domains where we're capturing the changes that are important to y'all as a community and how sleep disturbances look in Angelman syndrome. So difficulties falling asleep, difficulties staying asleep, and also disturbing the household with those sleep disturbances for your individual who has Angelman syndrome. In this case, this is a 7-point Likert scale, so 7 points is most severely impacted, 1 point would be not at all impaired. And so a reduction in that score is how we know we're seeing improvement. So we see a pretty early response of a reduction of about 1 point that's really sustained out to a year. And we're really excited to see that the sleep data is looking so clean and so consistent that, it's one of those things that can really impact the whole household. And I think that's represented in the quote here from one of our families. And then motor. We know that the kids have balance problems, ataxia, coordination issues. And so we want to make sure that we're capturing this as well. So we're using a similar tool as the ASA, the Angelman Syndrome Severity Assessment. We're using the gross motor domain in this case. So again, it's been designed to capture what are those things that we know are kind of unique in terms of motor impairment for the Angelman syndrome patients. And so this is things like improved ataxia, less gait disturbance, better balance, navigating obstacles or uneven surfaces. And so when I think about changes in those types of symptoms, I think about how much a world could open up for a kiddo who was previously a big fall risk, like you see in our quote here, and now they're not falling as much. So maybe it's a little easier to go somewhere like SeaWorld or easier to go on a hike as a family because they can navigate those obstacles a little bit better. And so these are things that can really open up their world. And then behavior. We know that this is another feature that can really limit what you're able to do as a family, because the behaviors can be quite disruptive and difficult to manage. I've sat with families and not been able to find a medication or a behavior therapy that could really get things to a level where it was easy enough to go out to eat as a family, even just to celebrate a birthday of a sibling. And so we know from the natural history data, these scores really don't change over the course of a year. But in our group that we were able to do this ABCC, a very classic measure of behavior that's been used for years in other similar populations as well, we see a reduction. And again, that dotted line is that threshold that we're crossing, that we don't believe this is by chance alone. We believe it's related to drug. And then finally, we can look at this all together, because again, each individual patient has a unique story, a journey that's their own, and a story that's your own. And we know that Angelman syndrome doesn't impact every family in the exact same way. So if we can look at the data as a totality and kind of a holistic, being able to look at the forest, we can see that in the columns, we have our symptoms, and each row is represents an individual participant from cohorts A and B, our expansion cohorts. And this is called the multi-domain responder index. And so what we do with this tool is we define what is that minimal important difference, that threshold that we need to cross that we think means we have made a clinically impactful change. And we call that green if we believe we've crossed it, if the numbers tell us, the data tells us that we've crossed it. Minimal to no changes represented in white, and anybody that's worsening below that threshold is represented in pink. And then they're organized by responder from high to low. And so what you'll see is that we get improvements across all these different domains and different individuals, and 79% of these kids have had a clinically meaningful improvement in at least one domain by a year. And so we're really confident as we're moving forward that we're seeing things that matter. So we want to look at it one other way. We've presented this data to you in October before, so we've shared this before, but I just want to bring it in one more time as we're looking at, again, what does all this mean? And so if you start to look at what are the new skills that these kids are acquiring over the course of the study, we have improvements in following directions, 1 and 2 steps, kids that are gaining words, word approximations, even some that are gaining multiple words or phrases. And we've got kids that are having reductions in their challenging behaviors, making it easier to go out as a family or even just have a simple dinner at home. And so all of these things add up to us having very good confidence that we have a drug that's making a change, and now we get to go test it in a phase 3, and we're really excited. But before we get there, we want to talk about safety. So in trials, we always monitor for safety. That's always going to be part of every single interventional study anyone ever participates in. And so you always monitor for adverse events or potential side effects or things that happen that we have to assess. Are these related to drug? Are they related to the study procedure? Or did they just happen by chance? So you'll see the most commonly reported treatment emergent adverse events here in the top. We've also had our adverse event of lower extremity weakness or leg weakness. We had our initial 5 patients who had a much higher dose and had this lower extremity weakness. We took that opportunity to learn, to grow, to change, and make a number of adjustments that have really reduced the number of times that we've seen that happen and have given us a really good understanding of how we can reduce that incidence going forward and manage it. Importantly, everyone has recovered completely. There's no sequelae, no long-term effects, and everybody continues to be in the study and is really doing well. So since those initial 5, we had 1 patient in cohort 7 and then 2 in our expansion cohorts. And again, we're confident that these kids are doing great and are going to continue on in the study. So we've had a consistent safety profile. We've got no new safety concerns, and we're heading into our phase 3. So with our sustained improvements out to 3 years where we have that data available, our consistent safety profile and consistent improvements across multiple domains, we're really excited to share with you our plans for the phase 3. Some of you may have tuned into our webinar. We go into a lot more detail there just due to time constraints here, but we'll share some of the highlights now. So it's going to be a global study. We'll have about 120 kids, and we're using the same population or planning to enroll the same population that we studied in our phase 1-2. You don't want to change too many things when you're going into your phase 3. So we're still going to study patients with deletion genotype ages 4 to 17, but remember we have Aurora, our other study where we're going to investigate in the other kiddos. So consider participating if your child is between this age range, has a deletion genotype, can walk independently, and has never received another ASO, and that you're willing to participate in all of the trial activities. We understand that, this is not just going to the doctor. These are protocols with a lot of extra assessments, and so we appreciate your partnership in that. And then our primary objective is going to be similar to the data I just showed you. We've got great confidence in what we've seen in our phase 1-2, and so we're going to continue to assess all these plus the other domains and assess them in multiple ways so that we really get a good understanding of what's that efficacy profile of GTX-102. And it is up on clinicaltrials.gov, so that number can be found here, and you can also find it for EU trials as well. Okay, so what can you expect? What is the trial going to look like? So we will have a screening and randomization period. It'll be a 1-to-1 randomization schema, so like the flip of a coin, whether you go into the GTX-102 treatment arm or the control arm. And we're using a sham control. When we were thinking through the design of this study, we understand that this is burdensome and that these drugs are delivered by lumbar intrathecal injection, so a lumbar puncture or a spinal tap. And so we did not feel that it was necessary to inject a placebo into that spinal fluid. That's a really special space, and we felt that introduced more risk than we needed to understand the safety and efficacy profile of our drug. So it will be a sham control. The treatment arm will continue through escalating doses up to our gold dose. And that double-blind year is going to be about a year long. At the end of that year, the treatment arm will be unblinded and continue to receive their maintenance doses over that second year. The control arm is going to have a mixture of sham procedures. It'll be a pinprick in the lower back, as well as some procedures where we will collect some spinal fluid, because we do think it's still very important to collect spinal fluid from patients who are untreated. So essentially like a natural history cohort, right, of getting this really important fluid. It helps us understand the safety, but even more than that, we're going to partner with FAST and ABOM and ASF to do additional studies and see if we can help develop biomarkers that are relevant to your disease, relevant to Angelman syndrome, that could help us understand this trial, but also potentially future trials. Thank you. So, we understand that this is not without risk, it's not without burden, no clinical trial is, but we want you to be as informed and empowered as possible. So, when the sham arm, the control arm, completes that 1-year participation, they will transition over to the treatment arm in the open label phase and get the exact same regimen that the treatment arm had the opportunity to receive in the first year. When everybody's finished their 2 years of participation, each individual will roll over into that long-term extension study I mentioned and continue to have access to GTX-102 if they so choose, where we'll continue to learn about the long-term safety and efficacy of GTX-102 in these populations. The double-blind period, it's important to know, it's been told to you a million times, I'm going to be the one last person this morning that tells you, it's so important to try not to guess, try not to push people to tell you which arm you're in. I know it's very tempting, but our goal is that you don't know, I don't know as the sponsor, your investigators don't know on site. There's only a limited number of people at the site who will have that information, and that's so that they can administer the different procedures between the two different arms and monitor the safety. but we're protecting as much as we can, especially in terms of our efficacy outcomes. So we really appreciate your partnership in this and collaboration. Okay, so again, escalating doses by intrathecal injection, some blood and urine tests, as well as that spinal fluid, which is so important and so valuable. Physical exams, developmental testing. You're going to do more Baileys. I'm sorry, I know that's frustrating, but it's an important tool that we need to understand what it looks like in this controlled setting. You're also going to do more questionnaires and surveys, which I know can be lengthy and onerous, but we really appreciate the information and your honest opinion. And then some participants will be doing EEG. We acknowledge that your group has done some really amazing foundational work on EEG in your natural history studies, even linking it to Bailey assessments. When I was in academics, I was referencing your work on other disorders. So you have done really incredible legwork, and we want to continue to learn from that in a clinical trial setting. It's a global study, and so we know that that's going to involve travel for a lot of you. These often are not sites that are in your backyard, and so we'll help support you find a trial site that's close to you once we're able to connect you with that trial site, and then also cover those travel-related costs to try to offset some of that. And we understand that it is time. You're giving us time, which is one of the most valuable things you can give up, and so we just really appreciate that. So, if you want more information, stop us, find us. We're excited about the program. We're happy to talk to you. We've got a wonderful team here that's at our booth. And full disclosure, those are the people that sit behind this email address. So when you are emailing these folks at trialrecruitment@ultragenyx.com, it is a real human who knows our program, knows us, asks me questions, asks our team questions if they don't know the answer, and they'll get back to you with as much information as they possibly can. But this is a very personal decision. You have to think about it. You have to talk to your family. You have to talk to your loved ones. You need to talk to your medical team at home also. Does this make sense for my child? And so we want you to have all of those conversations with the people who know and love you best. And then you can have conversations with us and ask us as many questions as you need. But we also want you to build relationships with those sites. And so we may sometimes direct you to the sites to ask some of these questions so that you can start getting to know them as well and their team, because they're going to be monitoring your kiddo and watching over your family for a while. Okay, so in summary, we've got exciting phase 1/2 data. We are showing a promising benefit-risk profile, and we're heading forward to our ASPIRE trial. ASPIRE is going to be a global study, and it's opening by the end of this year. And our intent is to have it as geographically distributed as we can so that as many patients as can have access to it, and we can get it enrolled as efficiently as possible. All patients will have access to GTX-102 from the clinical trial for the long term in the long-term extension study. And then we're exploring those additional genotypes, additional ages in our Aurora study with that planned opening in 2025. Okay, so with that, I thank you for your time, your attention, and letting me be here.
Are the doses based on weight? So will a 2-year-old and a 40-year-old get the same dose? And what is the highest dose that's been given, and what is the doses that are planned for the phase 3?
Great questions. So our doses for the phase 3 are based on what we've used in our dose escalation and dose expansion cohorts. For Aurora, we did make some adjustments. So we know that a 2-year-old is smaller than a 4-year-old, and so we made some adjustments to understand what can that lower dose look like. And so we have some plans. Nothing's quite finalized for the younger kiddos, but our intent is to use the same dose from ages 4 and up, and we're still working through the younger dosing.
For the Aurora trial, is that planned to be global as well, or is it just US based?
Nope, our intent is for that one to be global as well. So we're going through site feasibility now, and as soon as we have information we can share, we will certainly do that.
Fantastic, thanks. Goody.
Hi, my name's Charlotte. I'm from the UK. First of all, thanks so much for all the data. It's been really exciting listening to it, but hearing what that means in real terms for our kids as well and seeing the improvements that they've made is just fantastic. So I've got two questions. The first of which is, what do you mean by independent walking? Because, for example, my son can walk with a walker, but not without it.
Yeah, that's a great question. So it's not a firm definition. We are training our investigators to use their good judgment. We're using gait as one of our functional outcome measures. So understanding, what are their improvements in walking? So it's an important measure to be able to understand what's their baseline look like and how can they improve from there. But also from a safety perspective, do we notice any changes in their walking that may be an early sign that we need to think about that leg weakness? So really it's just, we wouldn't want a patient in our phase 3 study who is wheelchair bound or unable to walk most of the time. And so understanding what their baseline looks like is important. It'll ultimately be at the discretion of your investigator. So we give the guidance, they make the decisions. For Aurora though, we know that kiddos under the age of 4 don't typically walk or may not be walking yet. And so we are thinking through what are some of those other ways we can monitor for motor function as well as safety.
Brilliant, thank you. Mm-hmm. And then the next question is, obviously part of the next cohort is going to be placebo. So what does that look like for the parents in terms of at the end when you find out what your child has been on? Is there additional counselling support? Is there things to put in place for the obvious disappointment that is going to be there if you found out your child is not receiving the drug?
Yeah, that's a great question, and it's such a tricky one. I think in the ideal world, our regulators would trust your natural history data. We're just not quite there yet. And so we are in a space where we still need to do that controlled study to understand what those differences are. We will have a window of time between that crossover from year 1 to year 2, and that's when you'll be informed. And so we're still talking through what are the ways that we can support your community. We've heard you. We've had that feedback before. And so we're working with ASF. We're working with FAST to understand what that support could look like.
Over here. For the ASO design, do you have any baseline data with any polymorphism in your design in the normal population? Second, the plan design is intended to collect that data as part of assessment in case that individual has a polymorphism on the ASO design sequence side?
Are you meaning like in the non-clinical, the preclinical studies?
No, that's a possibility. Someone has one base pair difference in their ASO target, then your design, they are not going to, and efficiency going to be reduced or changed. They're different than other perfect design.
Yeah.
Do you have a baseline data for that design in the population? What's the frequency in the population, or do you plan to include this information as part of the evaluation study, collect a sequence for individual to evaluate that?
That's an interesting question. We are utilizing clinical genetic testing, so if that information's in their report, we've not done additional genetic testing on any of our patients that have been enrolled in the study to date.
I have one. Hi. My name is Lauren Selby, and my question is, and thank you for everything that you and all the pharmaceutical companies are doing for our children. We appreciate it. So if a child, just say generally, was in a trial and it no longer exists, and they had a washout period for over a year, meaning the ASO is no longer in the body, functioning not there, could that be a possibility of being in a new trial if they've had over a year of a washout period, which I know that was one of the criteria of joining ASOs, they had to have a washout period. Could that be something that they could do, even though I know you said that was something they can't?
They're still talking through what makes the most sense to support and potentially serve those populations, those families who have had exposure to other investigational drugs. There's a lot of clinical trials going on and in the pipeline. So this is a question that we know is going to be coming up, and we're thinking about it. We want to make sure we do whatever is safest And this is still early days for us understanding what do ASOs do and look like in kids with Angelman syndrome. So our first and foremost goal is that we don't expose people to unsafe things or unsafe plans, and so know that we're thinking about it and trying to decide what's that best place.